So now we can start talking about primary structure of protein. So we'll be talking a lot more about protein structure later on. But for now, and you may already know this, that proteins have 4 levels of structure. The primary structure, the first level, is really just the sequence of amino acids that make up the protein. So all proteins, by definition, are linear chains of amino acids. So when we have the sequence of those amino acids, we know the primary structure. And then later on, we'll learn that the protein itself folds up on itself, and it is stabilized by interactions between the backbone structure of the protein, and that's called secondary structure. And there's different types of secondary structure, like the alpha helix, and we'll talk about it and other types of secondary structures later on. And then those secondary structures can fold up, and you can have interactions between the side chains. Again, those hydrophobic interactions or ionic interactions or hydrogen bonding, all of those contribute to give you tertiary structure, and that's the ultimate 3D shape of a single polypeptide chain. And so some proteins simply exist as single polypeptide chains, and so that's the highest level of structure they have. But most proteins then assemble with other polypeptides to form larger structures and we call that level quaternary structure. So most proteins are actually multiple polypeptides that bind to each other, again, usually through non-covalent interactions, but they form large complexes. So we'll be talking about those higher levels of structure later on. For now, we're going to talk about primary structure, just the sequence of amino acids in the protein. Again, it's easy to describe the primary structure, but the question is, how do you find out what the sequence of a protein is? Nowadays, most proteins are sequenced or are not sequenced at all, but rather the sequence of the amino acids is just determined by the DNA sequence of the gene. So later on, we'll learn about the genetic code, and we'll learn that every amino acid is encoded by a set of three nucleotide codons that are called the genetic code. And so if you know the DNA sequence of a gene, you can directly figure out the amino acid sequence of the protein that it produces. But if you have a protein of unknown identity, and so you have no way to know what its DNA sequence is or the gene that corresponds to it, then you have to sequence that protein somehow. And there are lots of ways to sequence a protein. And in fact, nowadays, a lot of these things are automated. But again, if they're done at all, because protein sequencing has become less and less important as DNA or as genetic information has become more widely used. But it's still important to know how this is actually done, or how at least it used to be done. So probably the best known and most widely used method for sequencing a protein is called the Sanger method, developed by a guy named Sanger. And it involves basically breaking the protein up into fragments and then sequencing those fragments and putting them back together. So usually the first step of the Sanger method is to determine which amino acid is at the amino terminus. And to do that, you simply tag it, basically add a covalent tag to the amino group that will let you be able to tell which one it is. So, for example, this molecule, fluorodinitrobenzene, will react with the amino terminus of a protein and attach to it. There are other things like danzyl chloride, dabzyl chloride. Those are all molecules that have been used to tag the amino terminus of the protein. And then once it's tagged that way, then you can break it off and just look for the presence of that tag to figure out which amino acid is there. And then on top of that, or in addition to tagging the amino terminus, you can figure out the amino acid content by simply taking the protein and breaking it up into its individual amino acids. And this is done by what's called acid hydrolysis. So if you put most proteins into a low enough pH solution, the amino acids will break apart into their constituent amino acids. You'll break apart the peptide bonds through hydrolysis, and instead of a whole protein, you'll just have a mixture of amino acids. And then you can do other methods to figure out what those amino acids are. So that by itself, though, doesn't tell you the sequence. That just tells you what amino acids the protein is made of, but not what order they go in. So, but once you know what amino acids are in your protein, you can decide what cleavage reagents to use. So when we talk about cleaving a protein, it just means cutting it into pieces. And to do that, you usually use these enzymes called peptidases. And there's lots of different types of protein digesting enzymes. Most of them come from the digestive system. So when you eat proteins, they're broken down in your digestive tract. So lots of organisms have proteins that are dedicated to breaking down or breaking peptide bonds. And many of them are specific for particular amino acids. So trypsin, for example, is a commonly used one, and it targets either lysine or arginine residues on the C side. So that's what this means over here when we say that the target of this peptide is a lysine or arginine, and so it breaks a protein wherever there's a lysine or arginine on the carboxy side of that amino acid. And so each one of these enzymes or reagents has a different target that it reacts with. So once you know the amino acid content, you can pick one or more cleavage enzymes to cut into appropriate fragments. Then you actually sequence the fragments. So you can do this with if your peptide is short, you don't have to do the previous steps, but most peptides are kind of too long for this particular method, so you cleave them first. But the actual sequencing of the fragments, again, there are multiple ways to do this. This method is called the Edman degradation, where again, the idea is you add your peptide to a reagent that will bind to one end. So in this case, again, it's binding to the amino terminal. It's this molecule called phenylisothiocyanate, and under high pH, it will bind to the amino terminal. And then when you lower the pH, that will actually break the peptide bond between the amino terminal and the next amino acid in the chain, leaving the next amino acid with its free amino terminal. And then you extract the now newly formed molecule that contains phenylisothiocyanate on it, and figure out what the residue for that is. So once you identify that first amino acid, then you know you've sequenced one location. But your shortened peptide, the one that now is one amino acid shorter, has a free amino terminal, and it can go back through this cycle and be modified so that now the next base or the next amino acid can be identified. So it's this kind of cyclic reaction where you identify one amino acid at a time until you figure out the whole sequence. But again, you usually do this on short polypeptide regions. So you end up with these fragments that are sequenced like this. So we from this protein, we have 4 fragments, and the letters are the individual amino acids. And their order. So then you have to kind of put them back together again. You have to figure out how they went back together in the original protein. So once you determine that, so in this case, you learned from the original amino terminal labeling. Like, let's say we figured out that glycine must be at the amino terminal. So we look down here at our fragments, and we know, well, fragment 3 starts with a glycine. The rest of them don't. So that means that fragment 3 must be the first fragment in our segment. And then we can also look back at our cleavage fragments. And so again, if we use trypsin, so if trypsin was our enzyme that we use, remember trypsin only picks lysine or arginine residues. All the rest of these have so fragment 1, fragment 3, and fragment 4 have a lysine. So fragment 3, this K is a lysine, R is arginine, whereas fragment 2 ends in an E. And so that means that it does not have a trypsin cleavage point on it, so it must be the carboxy terminal. So now we've figured out the two ends. And then you look for places where they overlap. So if you can find the place where the other two fragments overlap together, you can figure out where they go in the whole sequence. So that's the basic steps of the Sanger method. Now, obviously, this is a pretty labor-intensive process because it requires a lot of steps, and of course, it requires a protein in large quantities. Most of these steps degrade or destroy the protein, and so if you only have a tiny sample of it, it's not a good method for sequencing. You need large amounts of it, of your protein that you're trying to sequence. So modern methods of protein sequencing are usually faster and require less quantity of protein to start with. A common technique is called mass spectrometry. And in this case, you take, this can be used for any type of molecule, but you pass your molecule, you sort of push it out through a thin glass tube, and as it goes through that tube, you apply a voltage to it so that the molecules get surrounded by this static electric charge. That comes out and goes through a little slit, and it's directed by an electrical field into a detector, and then the detector basically measures the mass to charge ratio. So by measuring that, you can figure out the molecular weight of every molecule that comes through. So you can do this with proteins. It doesn't work as well with whole proteins, but again, with individual fragments or individual amino acids, you can do this. So that's an easier way to identify amino acids because, again, every amino acid has a characteristic mass, a molecular weight. So if you know, if you measure the molecular weight of the amino acids in order, if you cleave them off in order and pass them through the mass spectrometry device, then you can figure out the sequence of the amino acids. A somewhat more sophisticated method for doing this is called tandem mass spectrometry. You're basically doing it, running your protein through the mass spectrometry system twice. So you run your protein through one mass spectrometry system, and then another, and then the proteins collide with each other and they break up into fragments. And then again, you have a detector that measures the mass charge ratio, and again, if you know the amino acid content of your protein, you can figure out from the mass from the size of those fragments what amino acids they were made of, and you can work out the sequence. So again, this is just a slightly more complicated, more sophisticated way of sequencing proteins. So again, you end up with kind of a graph like this that shows you different peaks for different amino acids. So what do you do with the information once you have sequenced a protein? Obviously, knowing the sequence of the amino acids in a protein will tell you something about that protein. It'll tell you some information about the physical properties of it. But another really useful thing you can learn from protein sequencing is something called bioinformatics. You can take large quantities of protein sequencing data, and you can compare the amino acid sequence of proteins to that of different species, and you can learn a lot about not only the protein, but you can learn about the species and in some cases the evolutionary history of different groups. So here, for example, this is called sequence alignment. So once you have two different proteins that are from different species, or the same protein from two different species, you can align them together. So that just means finding the equivalent protein in the other species and figuring out where they match. So here we have a protein from two different species and one species of bacteria, actually two different species of bacteria, and the kind of shaded regions are the amino acids where they match, and then any place where they don't match, that means there's a different amino acid there. So that can tell you something about the protein right away, because presumably the places where they match, in other words, the amino acids are the same, what we say is those sequences are conserved. So to say that a sequence is conserved that means that that region is must be important to that protein, because again, if these two bacterial species are not closely related, that means that their common ancestor probably lived perhaps hundreds of millions of years ago. So if these conserved regions, the implication is these regions haven't changed much in however long it's been since they split from each other. So that means that those regions must be important for the function of that protein. On the other hand, the regions that are different maybe can tell you how the function of that protein has been important for the evolution of that species. So you can look and see and say, well, if they're different, if this sequence, for example, here we have a gap. So sometimes this happens with alignments where you see a region where one protein has a set of amino acids present and the other one is missing amino acids. That doesn't mean there's an empty space here. So if you were to look at just the protein sequence from this species, this Bacillus subtilis species, assuming that's how that's pronounced, then the sequence would just go from this GDG to this TFE, and that would be the whole sequence. So it's not that there's a gap there, it's just that when you align them, there's a gap. And you can have multiple gaps. And so in fact, you have to be very careful to make sure you're really comparing two different versions of the same protein if there are large differences between the two. Now, another thing you can do is compare. So again, if you see certain sequences of amino acids showing up in a given protein across many different species, those sequences must be important. And in fact, when we see the same sequence of a protein showing up in many different species, we call it a consensus sequence. So a consensus sequence is just a region of a protein that is either, if not exactly the same from one species to the next, is very, very similar. And so this is just one way of depicting a consensus sequence. So this is a region of some protein. I don't know what protein it is, but they're looking at 13 amino acids within this protein. And so the amino terminal is on the left, the carboxy terminal is on the right. And for each position, the letter represents the amino acid that can be found at that position. And then the height of each letter tells you how highly conserved it is. So for example, for this protein, there's an aspartate always found at position 1. That's what the letter D stands for. Whereas other positions, there seems to be a lot more variability. So, if you look at, like position 9, there's at least 5 different amino acids that seem to show up there. And then some of them, there's so many that they're squished together, so you can hardly even tell what the amino acids are. But also it's written out at the top to give you a sense of all the amino acids that show up in that position. And again, this can tell you some important information. Typically, for example, look at position 8, there are 4 different amino acids there, but all of those have something in common. So isoleucine, leucine, valine, methionine, those are all non-polar amino acids. So that's pretty common too, where you may see differences at certain positions in amino acid sequence, but the amino acids that are there are all similar in property, because again, as we'll learn later, when the amino acid sequence determines the structure of the protein, but and if you change from one amino acid to another, so if there's a mutation that changes the amino acid sequence, that can change the function of the protein, but not all mutations are the same. So if you switch one non-polar amino acid for another, that will have less of an effect on the function of that protein, as if, let's say, you replaced a non-polar amino acid with a charged amino acid. And so the type of mutation matters a lot. But again, this can tell you something about the way this protein works. If you see certain positions or certain sequences that are highly conserved like these consensus sequences, that can tell you something about the protein. It tells you this region, because it's so highly conserved, it must be important to the function of the protein. And it can tell you again about the evolutionary history of that protein. On the other hand, you can look for sequences that do not have much in common between species. So if you do a sequence alignment and you see a region that basically has no conservation at all, meaning it's different for every species, then that is what's known as a signature sequence. So you can look for amino acid sequences, and if they show up in different species, but they tend to be common between or have more in common between two groups that are more closely related, then again, that signature sequence can be a clue about the evolutionary relationships there. So again, here we have one protein. We're looking at 6 different species. So 2 species of gram-positive bacteria, and then 2 species of archaea, and then 2 species of eukaryotes. And what you can see is that the signature sequence is absent altogether in the gram-positive bacteria and the gram-negative bacteria. So there's a gap there. But there is a sequence that's common to both archaea and eukaryotes. And if you compare the eukaryotes to each other, so you have one from a human and one from Saccharomyces cerevisiae is just the long name for yeast. And those two, the signature sequence is exactly the same. And meanwhile, the 2 archaeal sequences are somewhat more different, and but they're present, whereas they're absent altogether from the bacteria. And again, that reflects the evolutionary relationship. So we know, for example, that archaea are more closely related to eukaryotes than they are to bacteria. So that's why the archaea contain that signature sequence probably, and the bacteria don't. And we know that the 2 species of eukaryotes are more closely related to each other than either one is to the archaea. So that makes sense that you would see more consensus between those 2 than you would if you compare them to the archaea. So, but again, those signature sequences are able to be used to identify, what groups a species came from, or what group the protein came from. And then, speaking of comparing proteins, you can ask questions about kind of the evolutionary history of a protein and the function of those proteins. So again, this sort of depicts how proteins and species evolve over time. So let's say you have this protein X, and it's found in the genome of species A, and so we'll call it protein X sub A. And so many generations pass. This species it slowly evolves. Maybe it changes, but eventually it splits into 2 species. This is called speciation, when 2 species split into 2, and 1 species splits into 2. And the descendants from that speciation event again have 2 different lineages. But they still have that protein X. Protein X is found in both, but they're slightly different from each other. So protein X C is found in species C, protein XB is found in species B. It's the same protein, so it presumably does the same thing as it did in species A, or at least something similar, but the sequence of amino acids that make up each one may have changed. And again, that can happen due to just random mutations. But when you have 2 proteins that are descendants or are very similar to each other, but they're found in separate species, but their common ancestor came from the same species, we call those two versions of that protein orthologs. So for if you compare the protein sequence of, say, a human protein to a mouse protein, and they're very similar, and they're in similar positions, then you can conclude that those proteins are orthologs to each other. Presumably, or often, they would have the same function or a similar function in either species. What a protein does, again, depends on its structure, and so if you change the sequence, you're going to change its function slightly. But especially if it's a very important protein, there won't be much difference in the function between the 2 species. And then if you have another thing that can happen to a protein over time, or to a gene, is something called duplication. So when we'll talk about how genes get copied, how DNA gets copied later on, but what's supposed to happen is when a chromosome gets copied, all the genes in the chromosome are supposed to be copied and passed on to the next generation. But sometimes in the process of copying DNA, the enzymes that copy DNA screw up. And instead of just making an exact copy, you can end up with a copy where perhaps the one particular gene gets duplicated. So in other words, instead of just one copy of gene X, the gene that corresponds to protein X, you end up with 2 that are in tandem. That means they're in the same chromosome. or even a different chromosomes. It actually doesn't matter. But now you have 2 copies of that gene. Now, sometimes that makes a difference. Sometimes it affects the survival of that species. Sometimes it doesn't. But if it doesn't, and that individual with that mutation is able to survive and it reproduces and it passes on that now duplicated copy, both of those copies over time can mutate. And so you can end up eventually with 2 different versions of that protein in the same species. So many generations later, species B could have 2 versions of protein X, and one version may be so different. In fact, this kind of thing can actually lead to even more changes in the protein, because if protein X is important, you can't mutate it without and have the species survive or have the individual survive. So having a backup copy means that one of those copies, is now free to mutate and change. So when this happens, when you have 2 proteins in the same species that derive from the same ancestral copy, we call them paralogs. And so you can have 2 different proteins that may have partly overlapping sequence primary structure, but they can vary quite a bit and actually have completely different functions even in the same species. And so again, all of this information can be used to again, not just study proteins, but to study the evolutionary history of the organisms that the proteins came from. So comparing different proteins is a way to look at evolutionary history, because presumably every gene in every protein, not presumably, we know for sure, every gene in your body, in your cells, you got from your parents, and they got them from their parents, and so on. So if you go back far enough, you should be able to trace the lineage of all living things back to this last universal common ancestor. Now, we don't know what that was. Probably it was some sort of single-celled organism, but we can ask questions and look at where different branches came off and when. You can look for the number of differences between 2 sequences. So again, if you do a sequence alignment, and you can do this with DNA too. So this is actually, in fact, more commonly done with DNA than with protein. But you can compare 2 sequences after doing sequence alignment, and you can count the number of differences. So you can look and see how many amino acids are different, and you can estimate, how frequently that protein mutates, based on comparing many different sequences. And then you can say, well, if it mutates, so many times per million years, and we see this many mutations, then you just do the math and figure out how many years, how many generations it's been since those 2 species diverged. And so again, you can use this information to fill in these trees. You can also learn other interesting things. So for example, mitochondria and chloroplasts both have their own DNA and make their own proteins. So you can do the same thing. You can compare mitochondrial DNA or mitochondrial proteins with each other from different species. And from doing that, what we know is that those 2 organelles actually started off as their own independently living species of bacteria. And at some point in the history of eukaryotes, the ancestor to mitochondria got eaten, perhaps, or lived as parasites on some ancestor to modern eukaryotes. And then eventually they became dependent on each other. So the mitochondria became dependent on its host for nutrients and protection, and then the host became dependent on the mitochondria to produce ATP, and you eventually end up with modern eukaryotes, which cannot function independently of their mitochondria and vice versa. So they effectively are the same organism. But again, the mitochondria, by looking at their DNA and protein sequences, we can tell that they are actually related to bacteria, and then they became part of the eukaryote genome separately. Same with chloroplasts. Chloroplasts, which are found in plants and some other phototrophic organisms, are also organelles that have their own DNA. They also probably, we can tell from sequence data, bioinformatics, we can tell that they are descended from some sort of prokaryote that, again, either through as a parasite or other kind of symbiotic relationship, ended up becoming a permanent organelle in modern photosynthetic organisms. All right, so that is all we need to say about amino acids and primary protein structure for now. Next chapter, we will get into the structure of secondary and tertiary structures.